happy to be here in Heidelberg for the first time. So as Rebecca said, I'll talk about some of the work that we are doing on generative AI and how this can help us understand the physics of molecules. And so I've tried to make this a little bit more of a broad talk, a little bit, for some people, it might be a little bit basic in the beginning, but I just want to make sure that everybody is on the same page so we can understand why I'm doing what I'm doing and why it makes sense, okay? So, so why should we care about the physics of molecules? Seems like there's this thing over here that can move. So why do we care about the physics of molecules? So one thing is that, of course, that we're interested in understanding the mechanisms of molecular function. That's an example of this is the use of is most obviously illustrated by the work of, like, for instance, macrostate models, where we try to disentangle or make executive summaries of large MD data sets into these graph models that describe major configurational states, so the structural reorganizations, for instance, in large protein molecules and how rapidly they exchange with each other. And so here, this is an example from a recent work done by one of my graduate students in collaboration with a postdoc from Michael Sattler's group at the Technical University in Munich, where we were studying the pre-recognition dynamics of an engineered protein to neutralize a plant toxin called tichin. And so another reason why we would be interested in understanding the physics of molecules is to predict experiments. And so, for instance, in this work here, this Markov state model was also able to predict these or reconcile molecular simulations with in a mass experiments, so where we can measure directly the dynamics of exchange between different molecular configurations, and we could match align these pre-recognition dynamics directly with the experiments. But also in a completely different area, we're also interested in predicting these experiments, for instance, in the context of drug development. So in the one of my graduate students, Juan Diaz, who is also a student with AstraZeneca, so an industrial PhD student with AstraZeneca, he's working on, for instance, doing predictions of implicit of solvation of small molecules, which is a important task as part of drug development. And here we also, another problem that we would be interested in is actually to be able to predict these solvation-free energies. So how favorable, how much a molecule would like to stay in the aqueous solution. So what are these observables actually? So what are these? So so I'm just showing some numbers now. It's not really clear what they are. So some examples of what it could be. These observables, it could be like how many times per second does a drug bind to its target protein? How long does it stay bound? What fraction of time does my protein stay folded? Or how long does it take for a reaction to occur? How long does it take for a reaction to occur? Oh, yeah, 2 times. Okay, so we can illustrate this. This is a 4 a.m. slide. Sorry about this. Okay, so this, so we essentially we see there's a pattern emerging here. There's also something about quantifying things with respect to each other, with respect to time. And so one way of formalizing this is into a simple model is just to say, if we assume that there are two states of a system, A and B, and there's a model of interactions within this system that is described by these two sort of canonical states, A and B, that we call U of X. Then observables can be somehow related back to predicting the thermodynamics of the system. So that means the relative populations of the different states. Here they described as a free energy difference between the states, so delta G, for those who are familiar with this, or the kinetics or the exchange rates between the two different states. And this is all sort of governed in the end by the energetics of this function U of X of the system. So these two states could, for instance, be two metastable phases of a protein. They could be an unbound and a bound state of a protein and a protein ligand complex. or they could be one pair of states among many different possible states that are occurring. So this concept generalizes to arbitrary number of states in principle, but this is essentially the kind of things that we want to understand. So in the end, what these things are, there are some statistical averages with respect to the that are taken for the potential energy of the system. So this function u of x somehow encodes everything that we want to know about the system, but it doesn't tell us what states are interesting. So where does the randomness? So since we have statistical quantities, where does the randomness come from? This comes from the model that we use to evolve the system over time. So this is typically some variation of the Langevin equation. And the randomness here comes from, so what the Langevin equation describes is the displacement of atoms over time. And it can be in its simplest form written on this way. So the displacement of atoms over time is dependent on the forces that the particles exert on each other. And then a random term, which is just basically modeling how the system is in connection with a heat bath. So a lot of random particles that are kept at a fixed temperature. Okay, so this is where the randomness comes from. And so if we roll this out, so if we apply this simple equation, so a slightly more advanced version of this equation, but conceptually the same equation to run a simulation, we can get like these fancy movies like this, where we see protein binding and unbinding. So we can really quantify these processes with statistical significance, so we can compute these observables. So this is an example of then drawing samples from a statistical distribution, and then we can compute averages from this distribution, and that then correspond to what we observe in experiments. And so the outcome of this tour de force in molecular simulation was this model of the binding mechanism of these two small toy proteins of limited, I would say, biological significance, but still an interesting case, because this protein complex is a very low, so it's an ultra low affinity complex. So the binding is very transient, and the bound state is highly disordered. So it was extremely tricky from a modeling perspective to get this to work. And so an example of the simulation effort that went into this was it took around 1.7 milliseconds of cumulative simulation time. So anybody who has run a molecular dynamics simulation knows that this is a fairly long time scale for molecular simulations. And with the throughput of 300 nanoseconds per day at 250 watts, it's approximately 10 to the 11 joules of energy, which is about the amount of energy I spent coming here today. So, but it's also the equivalent to what you would typically do if you want to quantitatively study sort of a system of around 100,000 atoms with molecular dynamics. To quantitatively sample the system, you would need to invest this amount of energy. And so this is clearly not a scalable effort or scalable endeavor to do if you want to study a lot of different molecular systems, like you want to study a protein and then you want to study a mutant of this protein. So you want to understand what is happening, for instance, in a disease. So this will not be a scalable way of doing this. So as I was saying before, and now I'll get a little bit more technical, that these things that we measure in experiments, they are essentially what we call functions of state. So given our potential energy model here, it's a slightly different potential energy model, and it's here the x-axis is supposed to show the different monomeric states around this methyl group here that is centered on caffeine, and I believe there will be some coffee after the talk. So when we then do a simple transformation, or we do a transformation of this potential and specify a thermodynamic state, so for instance, the temperature, pressure, chemical potential, then we get an ensemble. So this is this randomness element that I was talking about before. So we get a statistical distribution instead of just a potential energy. And then what the observables are the averages with respect to this. So this is just a fancy way of writing a weighted average, where the P of X here is our probability distribution that is distributed according to the interactions of the physical system. Likewise, there are other observables that are time dependent, and they depend on the time evolution of the system. So here we invoke the Langevin equation again. And the outcome of this is that given we start in a single state, so say in this yellow state over here, x0, then the distribution of outcome states. So if we run infinitely many simulations that start in that state, they will have after a certain time t, there will be a distribution of possible outcomes. And this is what is shown here in the magenta distribution. And this distribution also gives rise to a family of observables that we can call, for instance, time correlations or autocorrelations that are very commonly measured in experiments. For instance, in the mass spectroscopy, many of the biophysical spectroscopies, they rely on the measurements of these types of quantities where F and G are some, for instance, physical phenomena typically related to absorption or exchange of energy with some sort of probe, so an experimental probe. And then we have some intrinsic physical properties. So we have the equilibrium distribution P of X here, which is the same as over here. And then we have the transition probability, which is the outcome of running these infinitely many simulations for t steps or for time t starting in the same initial condition. And so in principle, this seems like a very feasible thing to do for a one-dimensional system. So you can just run a lot of simulations. We can calculate these averages, but it becomes very tricky in general to solve this in high dimensions. And so what we rely on instead is doing Monte Carlo estimations of these integrals. And what this means is essentially that we draw a lot of samples, statistical samples from the distributions. So either from this distribution or from this distribution, and then we compute these functions of state F and G, and then we get our observable, for instance, the stability of a protein, so how long time it stays folded. That will be the fraction of time. So if you run a long simulation, it will be the fractions of states that are staying in the folded state and the fractions of states that will be in the unfolded state. That will give you the probability of being folded. Okay. And here it will, for instance, be like an observable that will be related here for the time will basically be you will correlate a bound state from and an unbound state will have a 0 and a 1, and then you will average this over time, and you'll see how this function decays, and there will be a characteristic bend in the curve when you reach the characteristic time scale of the binding or unbinding event. Okay, this Monte Carlo estimation. So the critical point here is that this relies on you drawing statistically independent samples. And this is not what happens when we run these regular simulations. We generally don't get independent samples. And the reason for this is that we solve this equation, a Langevin equation, with an algorithm which is called forward Euler, or some variation of this, which is basically just doing a discretization of the time. So instead of having a continuous time equation, which is this one here, we basically just say, okay, we don't know, we cannot make an infinitely small time step. So instead what we do is that we make a small time step on a computer that we call tau. And then we basically just do a numerical integration of this equation. So we basically just plug in the numbers. So we say, okay, we are currently at this point, and or we want to go to this point, and we are currently at this point, and so that should be equal to this, like a small taking a small step along the forces, so this gradient of the potential energy, and then giving the molecule a small kick with a variance which is proportional to the time step that you take. And so the outcome of this in practice is the following: is that if we have our potential energy function here, that is this blue in the background, taking a single step where we, for instance, start here, the outcome distribution will be very closely peaked around so we will in practice not move very far when we take a single step. Okay, so it's clear that if we then try to correlate the system state as a function of time, what we will see is essentially this. So the orange line, that gives us, this is what we get out for when we are looking at correlating these the trajectory of this system over time. So if we look at time t plus tau, t plus 2 tau, 3 tau, 4 tau, and so on. That's what we look at here. This k is a multiple of tau. And on this is the correlation coefficient between the system state variable at the initial condition and at the final state. And so what we see is that there's this very slow decay to 0. And it's not until we actually cross over this barrier 0 here that we actually have an independent sample. So there's a huge effort, simulation effort associated with drawing just one independent sample. And it's related to this quantity here, which is an eigenvalue. For those of who are familiar with Markov state models, it's related to the leading implied timescale of the Markov operator. Okay, and so essentially, this is a long-winded way of saying that if we just run regular simulations and calculate statistics from these, our estimators will be biased. We will make systematic errors in our averages and in the rates that we try to predict from the model. So essentially, the problem that we can see is with the current MD simulation tricks or methods that are out there is that there are two main limitations. The first one is this timescale separation. So this problem is that it takes a very long time to globally mix, so draw independent samples. So we have to do on the 10 to the 9 to the 10 to the 15 steps of integration to draw one sample. And if you want to compute many different and observable, then maybe we would need 10 times that amount of sampling statistics to get a statistically robust computation of these observables. And likewise, evaluating the potential energy takes some time. So there's been specific efforts in making this this part more efficient a lot with machine learning. I'll not mention any of the work that we've been doing in this area, but I know, for instance, Frauke has also been working in this area. And it's a very exciting and very sort of like one of the areas where machine learning has really had a big impact in the molecular sciences. But this is the main problems here. So we have to take a lot of steps, and each step is expensive, okay? But in the end, what everything boils down to is that, so from a machine learning point of view, is that, okay, this is all great. So we have these problems, but what is the core of the problem? What is the core of the issue we need to resolve? And it's essentially to draw samples from two different types of distributions. One of them is the Boltzmann distribution, which is this distribution here. It's not normalized, and over here the transition density, which is then specified by some model of the dynamics, for instance, the Langeban equation. This is essentially the two things that we have to do if you want to solve the so-called sampling problem, which is this problem of drawing statistically independent samples, right? And so, what are the opportunities for machine learning in and AI in this area? So one of them is this building circuits. So that's approximating this probability distribution that we need to iteratively simulate with some machine learned model that is very easy to sample from. Another thing that we can do is model reduction. So that's, for instance, coarse graining, reducing the system down to more manageable systems that we can then simulate faster. So that's more in the other direction where we try to make each step faster. And then there's also the other thing, another thing that I briefly mentioned before is this making the accurate physical interaction models faster. This is a different strategy. So if we think about a very simple way of building a circuit for molecular dynamics simulation. So instead of doing a lot of or doing one long simulation, you could imagine that you have some initial structures here, these magenta points on the line. So you have, for instance, gone to the PDB where you found a lot of protein structures, experimental protein structures of the same protein, and they happen to be slightly different configurations. And then you roughly group them into 3 different groups. And then you start simulations, and you see on all of them in parallel, so they run in parallel. So you're sampling, you're not relying on sampling all the transitions between all these states in a single simulation, but you are then parallelizing the sampling process across multiple different workers. And so in this way, you can collect sampling statistics of the transitions between these different states. And this is what you can collect these statistics into these transition count matrices that you can then row normalize, and you can get a transition matrix out, which is exactly what we call Markov state models. So these models, they are very simple, conceptually very simple, practically very difficult to work with for everybody who's worked with them on any systems that are larger than the common model systems that we use in the simulation field. But the really nice thing about these is that you can directly relate these matrices from this matrix here. We can directly relate these to these observables that we are interested in. So for instance, the binding, the affinities of drugs to proteins, the stabilities of proteins, and so on. So we simply take this matrix, we do an eigen decomposition of the matrix, and we can express these expectation values here. So these averages, we can express them as simple linear algebra from this matrix. And so this is really an extremely nice property, but of course this has the limitation that we need to have sampled all the transitions between all the states. So in order to populate these transition probability, or these transition counts here, or compute these transition probabilities, we need to actually see these transitions between the states. And if we don't do this, then the model will not be able to make any predictions. So it's more an analysis technique rather than being a predictive technique, okay? And so to address this problem, I had a grad student, Christoph Kohlhoff, who was working exactly in this setting. So we will imagine that we have this toy system here, where we have we have two metastable phases that are separated by a very high free energy minimum. We have collected simulation statistics that are started in this state and this state with black and red, and then in this state over here with orange trajectories. And we are collecting that statistics into these transition counts. And the unfortunate case is that we haven't observed any transitions here. So we will get a transition count matrix, which is block diagonal. So that means that we haven't seen this transition over this barrier. And so if we just use the traditional techniques to address this problem, we would get what is called a disconnected Markov state model. So that is basically a model that describes the internal dynamics between these two states very well, and inside this state very well, but it doesn't describe this transition. So you get two models of the systems, or two different parts of the system, but not a global model of the system. So we cannot predict anything about the relative free energies of these two metastable phases, nor the rates of exchange between them. So what Chris set out to do was basically to turn the problems on its head. So instead, we know that we can observe these transitions between these states experimentally. So we can go to an experimental lab and say, okay, we have measured. Do you have a measurement of this observable? And then we can try to say, okay, what would these zeros have looked like if we had had an experiment? And so this comes down to enforcing an additional constraint on this statistical estimator, and then you can actually get a connected Markov state model. Great. So Chris developed this idea, and it's basically quite technical. It was a big technical detour, but in the end, we came up with this estimator, and it relies on some fancy optimization techniques. But the nice thing is that we can systematically bias a lot of well-known standard data sets. So for instance, this BPTI data set, we can remove transitions between these. So we have, it's characterized by rotameric flips in these two cysteine bridges, so these yellow regions here. And we can systematically remove all simulation statistics that connect these two states to these two states. And then we can try to see if we use experimental data, can we recover that connection? And it turns out that we can. So here's a graph that describes the exchange between these different states. Here, the colors, they correspond to the different states. And the transition states that have been recovered are here shown with the dashed line. And we can see here that we nicely match the experiments. So this the green is the disconnected dynamo model, and the red is the dynamo model that's not been disconnected. And then we have an experimental rate, which is exactly on top here of this coinciding with the slowest rate here. And we also see here that when we predict the experimental data, that we also get a very nice match. So these are, again, NMR relaxation dispersion experiments. So they measure the slow exchange between, so a time correlation function of exchange between different configurational states. And so this was not only working on toy system, but it also something that we deployed. So this is something that I mentioned early in the first slide of the talk, where we had a similar situation. So we had this very slow exchange between two different configurational states, one that was binding this plant toxin and the one that was not binding the plant toxin. So we could use this approach together with the experimental data that they had to build a model that was connecting this state with these two states over here that was otherwise not connected because they exchanged on millisecond timescales, which is not something that was feasible for this project to run simulations for. And so what we did was we compared it against of course, the training data. It matched very nicely, but we also afterwards we looked at the structural states that we had generated, and we saw that there was a contact that was formed between two residues, isoleucine 97 and valine 84, that was formed in one state but not in the other state. And we indeed saw that there was a very weak NOE, which is an NMR measurement for distance, that was present in one state that was removed when so there was supposed to be in the unbound state when this lid here was closed down, but it should not be there when the drug or the toxin was bound to the protein where the lid was moved out of the way. Indeed, we saw that it disappeared in the case of the toxin being bound. So we could directly independently confirm the conformational exchange that we saw in the model. Okay, so this is maybe a little bit so far been a little bit disappointing for people who came to hear about artificial intelligence. I've just been talking about Arc models, but I thought I would motivate everything a little bit with this work, because all of these models, they work very nicely when conditions are right. And one of the main culprits of why these kinds of methods, they really fail, is because we have to somehow decide on what states are relevant. We have to discretize the system. You can imagine that if you want to discretize a very high dimensional system, it becomes very quickly very difficult to do that in an arbitrarily optimal way. And every time we make a mistake, you will get a systematic error that we cannot control. We can just know that it's there. We don't know how large it necessarily is, but we can systematically optimize it. But there's in principle no end to how much we can optimize this, and we never know if we're actually done or not. And so one way of thinking about this is then, can we avoid discretization altogether when we build surrogate models? And so one way of doing this is what we did with the Boltzmann generators. And here we see the problem of modeling the probability distribution here of the equilibrium distribution, so the Boltzmann distribution, not the transition probability distribution, as a problem of changing variables between a distribution that is easy to sample. So for instance, a normal distribution. For the normal distribution, we can draw independent statistical samples with very efficient algorithms. There are even like fundamental algorithms that are in all numerical computing software packages out there that can draw high-dimensional independent Gaussian samples. And so to transform samples that are drawn from this into a space that aligns with the configurational space, so for instance, of protein structures. And so we learn a function that changes a variable that is a random, so Gaussian blob, into a highly structured protein distribution of protein structures. And so for these methods to be generally useful, so we can actually compute things quantitatively from experiments, then we need to be able to evaluate the sample probability. So we need to be able to compute what is the relative probability of two different states. Of course, we can always evaluate, or we can draw, if we can draw samples, that's useful, but it would also be useful if we could then say, okay, we are modeling something, and there would necessarily be an error that will be introduced when we make a model of something. And so can we actually control this error and minimize it, or recover unbiased estimates when, and the only way that we can provably do this is by asymptotically is we can evaluate the sample probability. Okay, and so I'll just give you a brief primer on how these models work, or the types of models that we work with currently, how they work in practice. Is that this function f, this has been a bit of a magic thing so far, but essentially you can think of it as an ordinary differential equation, and it's parameterized in such a way that the solution, so when we follow, if we start at time 0 from a normal distribution, we draw a random sample, we integrate the ordinary differential equation, we follow the integral curves until time 1. The idea is that when we do this for infinitely many samples, then the distribution, the data distribution should follow the outcome of doing this integration infinitely many times. Okay, and so what we do is that we parameterize this ordinary differential equation using artificial neural network. So there's a deep neural network that is modeling how these dynamics, or how these samples, this should move from this Gaussian distribution into this highly structured distribution. And examples of this, so one example of how this looks like is that you go, for instance, from a uniform distribution to a bimodal distribution on the circle. It could look something like this. And examples of models that use this type of idea is diffusion models, so like stable diffusion and DALI and so on, and normalizing flows, or continuous normalizing flows in particular. And so these models are extremely powerful, and they're also sort of conceptually very nice, because they allow you to make these flexible and invertible transformations between different configurations. And so what we can do, for instance, so one thing that one did in terms of the prediction of solvation-free energy was to parameterize a lot of small molecules using a separation of variables into local harmonic degrees of freedom, so angles and bonds. They are sort of not moving very, they don't have a lot of anisotropic contributions to them, so we just approximate them as being static in the model. And then we model all the torsion angles of small molecules using some internal coordinate representation. So we just have this model of these angles that are rotating around, so two planes that are with respect to each other. So if we look at these orange circles here, they indicate the torsion angles. And if we collate them all together, then we'll represent the configurational state of the molecule on a hypertorus. So if there was only 2, it would look something like this, and we would transform some distribution into another distribution using this formalism that we talked about before. And so the idea was basically, okay, we say the artificial intelligence model is sort of ignorant of the fluctuations, of the fast fluctuations, but the slow fluctuations that are in the torsion angles are modeled explicitly. And then we use this regular empty simulations to model the fast dynamics. And so the way that we implemented this, we use this to predict solvation-free energies of small molecules. And the way that it works is that we use this so-called Boltzmann emulator to sample initial conditions, so sample where we should start the simulations. And then we run a lot of simulations in parallel with standard molecular dynamics. We do a reweighting in this reduced space, and then we can recover these solvation-free energies. So this is essentially here, this is all on molecules that were not seen, the model had not seen when it was being trained, but it was models nevertheless that were accurately, or molecules that had their observables accurately predicted. All right, so and so I'm getting to the point where we are now also trying to, or we were encouraged by these results. So we were also thinking about how could we move this to also predict dynamics. So originally I was motivating this move towards continuous variables with the state discretization, but I moved back to only sampling the equilibrium distribution, which is still interesting, still not a completely resolved problem, I would say. And so, but we could also still look at whether we can sample the transition probabilities. And the way we would then reformulate this model would be into a conditional model. So we again start with a normal distribution in some abstract state that we can easily sample from, and then we make this transformation depend on some initial condition and some time horizon n, which is some number of steps of some discrete time that we're interested in. And then the outcome is, again, a hard to sample distribution. And the one way to look at it is this, that if we would run this yellow distribution, which is our initial condition, so for instance, a single state, we would run it with a lot of small steps, or we could run it with arbitrary long steps, so with the black lines here. And then we would see how the distribution would change over this time horizon. So this transition, so we would be able to directly sample these outcome distributions as we want. This is what we call implicit transfer operators. And the way that this works is essentially the following. So if we imagine that this is a molecular dynamics trajectory and there's some arbitrary time axis here, and we just assume that this is a very long, very difficult to do simulation, then what we are doing when we are training these implicit transfer operators is that we are deciding on some initial state x0 here, and then we are training it to predict pairs of this initial condition, so the start point, and then a lot of to predict this endpoint here, which is the end of the arrows here, to given the initial condition and the time horizon, so how far away in sequence they are from each other. So this could be arbitrarily long things. So this is what we do. We train this on molecular dynamics data. So we first have to collect some data, and then we train the model. But then in comparison to what we do with standard MD is that we would zoom in on one point here, and then we would do one step here where we would basically not do any movement at all. So instead of being limited to just doing steps like this, we can now do arbitrarily long steps. And so we have, of course, done this on small molecules to start with. So this is alanine dipeptide, which can be described by these two torsion angles here in this free energy, this two-dimensional landscape. And so what I'm showing here is the transition densities, these transition probabilities that are modeled by the ESO model when we start in the red spot here. So the contour lines are what the generative model is capturing, and the blocky histograms in the background are what we get if we run very long MD simulations on the same system. And what I'm showing here on the following two plots are marginal. So when we project on to the x-axis and y-axis respectively, and we get these two things. And what is on the labels here is how long into the future we are predicting the endpoint. So as an example, in this upper panel here, we are starting in the red, we are starting at the red circle. We are starting infinitely many simulations, let's say. And then we let them run for 8 picoseconds of simulation time. And then we look at the distribution of the outcomes, and they fall like this. And we can see that qualitatively they match up very nicely. And also when we go to longer and longer time scale. And so one thing that is interesting here from a theoretical perspective and from a modeling perspective is that I'm here showing three different colors in these marginal plots here where we are projecting onto the X and Y axes. And so there's the blue line where we are doing a direct step from the initial step in one step to the final step. And then we have one where we are doing what we call nested sampling, where we are splitting the step, the model into doing 10 smaller steps, but they in aggregate sum up to the time scale that this single step does. And so based on the basic theory of molecular dynamics, these two things should be in distribution equivalent to each other. And we don't enforce this in our model, but we see that even despite that we shouldn't have this property exactly fulfilled because we are introducing some force grading, we see that indeed it does this. It does learn to be self-consistent, at least approximately. Okay, so this was a very nice sort of proof of concept, and we moved this up to also sort of mini proteins. So for instance, this was this time scale I was looking before. Here I'm showing some blue and orange curves that are indicator functions of whether a protein is folded or not. And this is for this small mini protein chignolin here. Here we have force grained to only see alpha atoms. So there's only one particle per amino acid for the protein. And when the region here, the protein is unfolded, and when it's sort of flatlining, then we know that it is in the folded state. And here there's a time trace here of a simulation that is done with MD, so that is run on the Anton supercomputer in Manhattan for a couple of days, I believe. And this is run on a single GPU for about 15 minutes. And we could so the cumulative simulation time here is 106 nanoseconds. The difference is here that they have to do they have to integrate with 2.5 femtosecond time steps, and here we can directly sample with 5 nanosecond time steps. So we can calibrate the steps of simulations with the time scales that we're interested in studying, rather than sampling these uninteresting events as the fluctuations within the protein. So here's an example of the simulation done with the generative AI model. And of course, if you don't know what it's supposed to look like in a regular simulation, it's basically indistinguishable from the reference. And it doesn't only sort of look okay, but it also quantitatively predicts for several proteins the free energies of folding and the average folding time and unfolding times, at least on the same. It gives the right order of magnitude for all of these values. So this is, of course, very encouraging, right? That, okay, we can recapitulate the statistics found in complicated simulation statistics, even when we are very far out in the tails of the distribution. So we are really trying to predict things that have very, very small statistics in the training data. And so the question becomes, okay, we can do this, right? But it's also 100 times more expensive to do just one step compared to doing one step of MD. So we have to be at least 100 times faster. So we have to be at least able to take 100 times larger time steps. But so we did a survey on this. So we looked basically on how fast, so how many steps, how many time scales of simulation time can we do per second of standardized computational resources. But this is for a 304 atom system estimated. So with the state-of-the-art CPU algorithm, we will get this throughput here, and with a GPU, we can get a significant boost with standard numerical algorithms. And here, this is what we can get with ETO, the equivalent. This is with just the current proof of concept state that we are in. We get a 5 order of magnitude boost in the throughput of simulation statistics. Okay, you can still be skeptical at this point, right? Because, okay, we are collecting a very large data set, and we are training a model on this data set, and then we are recapitulating all the statistics from the same data set. So that's not very interesting, right? We're just basically learning the same thing that we already knew. So is this really actually useful scientifically, or is this just a curiosity that is only of interest to computer scientists? And I would argue that we are getting to the point where it's getting interesting, because we can also, because this model is not relying on discretizing the states into these different configurational states that may not be shared between different molecules, but it's learning these continuous transformations that are agnostic to such states. Now we can train these models in a transferable way. And so here, this is a bit of a busy slide, but essentially what I'm showing here is these free energy landscapes. So we are showing different the probabilities of, so when there's a higher, stronger color here, that means that there's a more probability in that area. So there's a higher free energy, lower energy lower free energy in an area with a lot of color. And when it's white, there's no probability density at all. And here I'm showing predictions for a lot of three small molecules here that have not been seen by so it's a model that is trained on a lot of small molecules, but there's a number of small molecules that we have kept out for testing. And then so we are basically simulating them, and we're trying to recapitulate, compare those to the reference simulation data that we were collecting, but not showing to the model. And it's actually able to nicely capture all the metastable phases of these molecules, which is then an indication that we can actually, we're at the stage now that we can collect data on multiple different molecular systems. We can train a model, and then we can amortize the cost of collecting the training data by the speed up that we get from running the ETO simulations compared to the regular molecular dynamics simulations. And so it's not only the thermodynamics that are right, but we also get the exchange rates. So this is measured by something which is called the DAMP score, which measures how accurately in percentage we get the rates of exchange between the different states. So we both get the thermodynamics right, but also the kinetics right. And another idea here is that another interesting observation is that we can also actually compare the energies of the molecules. So we show in this is energy on the x-axis, and this is the probability density on the y-axis, or frequency on the y-axis. And what we are showing is essentially that there's a high overlap in energetically, so there will always be some errors that we introduce, as I was mentioning before, because we don't get the details right, but we actually get quite close. There's a the point here is that there's a high overlap between the energy histograms here. We can also scale this to slightly larger systems. So this is tetrapeptides. So this is a small, you can say, mini proteins with only 4 amino acids each. This is again 3 examples of many among many examples that we have now, where we can recapitulate the training data almost perfectly. At least we get the right modes. Here it's a little bit more washed out. You can see that there's some details that are being blurred out in the model compared to the reference, but it's still pretty impressive. And again, the dynamics are very nicely captured, even if we are doing all these approximations that we are. And so this would be wholly sufficient to accurately predict a lot of experimental observables if we had for these systems. Of course, we're not there yet. There are still examples where we dramatically overpredict the number of states that we have, or the variance of the states. As for instance, in this case here, where we should have these 8 states, but instead we have this more or less continuous blob of states. So we cover all the states, but we are overpredicting the transition regions in certain cases. And so a remaining challenge is, of course, to systematically overcoming this and understanding when these types of problems occur, coming up with systematic tests for this when we are very far, when is our model very uncertain about what it's predicting. All right. Let's see. How am I doing on time? So I'll just here at the end switch gears a little bit, where I was working with 2 of my master students, Selma and Wei Long, on how we could generalize this concept to not only so because we can learn these general transformations between the random variables, we could also think about transforming between different thermodynamic states. So instead of transforming from some abstract distribution that is normally abstract space that is normally distributed to a physical state, we could also transform between 2 different physical states. And so an example of this would be that we have an ensemble, a physical ensemble at 1 temperature, and then we transform it to another ensemble at a different temperature. And so what Selma and Wei Long did was to basically set up a training procedure to do this for many, where we basically give in the input of the 2 temperatures, and we learn a generalized transform that can transform between any 2 different temperatures. And the idea with this is that if we want to study a system at many different thermodynamic conditions, we could train a model on a subset of these conditions, and we could amortize the cost of simulation the other states by making predictions. And so this work is very conceptually related to Boltzmann generators, and it's actually a special, or you can say a instantiation or a specialization of the case that was a special case that was discussed in our original paper from 2019, where we were training 2 different independent Boltzmann generators and using them to predict energy difference between disconnected states because of their connection to a shared reference state. It's a bit of a deep cut for those of you who know about this sort of like additional experiment, but this is essentially a way where we can make use of this insight also in this context here to formulate these transformations in 2 different ways, either as a direct transformation or through a transformation through a shared reference state. And so what we do, so the way that we do this, so we do this for a number of small systems in our preprints, and now also, for instance, this small molecule here, where we see the transformation from one thermodynamic state to another thermodynamic state, and we can accurately predict the change in the free energy as a function of temperature using these different methods. Great, and we also maintain relatively high statistical efficiency when we do this. And so we teamed up also with Felix Nyske from Magdeburg, who is an expert in Koopman operator methods and theory, to extend this so we could also predict kinetics based on some idealized assumptions of how the equilibrium distribution is related to the generator of the dynamics. And so we could also actually predict the rates of exchange in these small molecular systems as a function of temperature by only sampling the equilibrium distribution. And so in the summary, I would like to say that yeah, I hope that I've convinced you that we can build now generative circuits for molecular dynamics. I can predict thermodynamics and kinetics almost quantitatively. I would say the right order of magnitude, we're there. It's robust mod reduction. So there was this point that I was making about that when we are removing some degrees of freedom, we are removing solvent, we are force graining the system, it still has the behavior that we would expect for a full resolution model, at least approximately. Generalization remains a bit challenging. There's still things that we need to address there. And the computational efficiency is still limited. I mean, we have gotten 5 orders of magnitude, but I mean, there's still a large number of systems that we need to cover, right? So the 5 orders of magnitude may seem like a lot, but in the grand scheme of things, it will not be something that will be able to replace high throughput experiments anytime soon. So we would need to probably gain another times the same for this to be truly useful. Going forward, we need to, I think, get more data from different sources. I think that's one way of doing it, and also making the architectures more efficient so we can close the gap. And so with this, I would like to thank my group and WASP, BACT, and the Wallenberg Foundation for funding. Especially, I would like to thank the people who've been doing the work, which is Matthias, Juan, and Chris. They have been the main leaders of the first part, and then Weilong and Selma, who did the final work. And so I'm happy to take any questions. Thanks for your attention.